Approximately 45,000 years ago, Neanderthals and humans coexisted in close proximity within northern Europe. Greetings and salutations to everyone. Today's video focuses on a recent genetic analysis conducted on bone fragments discovered at an archaeological site in central Germany. The results reveal that modern humans had already migrated to northern Europe approximately 45,000 years ago, coexisting with Neanderthals for a significant period before their eventual extinction. This discovery solidifies the status of the site near Ranis, Germany, as one of the oldest known locations of modern human Stone Age culture in north-central and northwestern Europe. The fact that Homo sapiens and Homo neanderthalensis cohabitated aligns with genomic evidence suggesting occasional interbreeding between the two species. Without further delay, let us begin our discussion. Through a thorough genetic examination of bone fragments discovered at a central German archaeological site, it has been definitively proven that Homo sapiens, or modern humans, had already migrated to northern Europe 45,000 years ago, coexisting with Neanderthals for several millennia until the latter's eventual extinction. The research conclusively proves that the location near Ranis, Germany, renowned for its meticulously crafted leaf-shaped stone tool blades, stands as one of the earliest documented sites of Stone Age culture by modern humans in the regions of north-central and northwestern Europe. The coexistence of Homo sapiens and Homo neanderthalensis is supported by genomic evidence, indicating sporadic interbreeding between the two species. This further fuels the speculation that the arrival of modern humans in Europe and Asia approximately 50,000 years ago played a role in the demise of Neanderthals, who had inhabited the region for over 500,000 years. Today, in the esteemed journals Nature and Nature Ecology and Evolution, a trio of papers presents a comprehensive account of the genetic analysis, as well as the archaeological and isotopic analysis and radiocarbon dating of the Ranis site. The leaf points, also known as stone blades, discovered at Ranis bear a resemblance to stone tools found at various locations in Moravia, Poland, Germany and the United Kingdom. These tools are believed to have been crafted by the same cultural group, known as the Linkombian-Ranisian Jersmanowitian (LRJ) culture or technocomplex. While the Ranis site had previously been dated to be at least 40,000 years old, the absence of identifiable bones left uncertainty regarding whether the creators of these tools were Neanderthals or Homo sapiens. According to Elena Zavala, a Miller Research Fellow at the University of California, Berkeley, and one of the four first authors of the Nature paper, the recent discoveries provide evidence that Homo sapiens were present in Europe 45,000 years ago. These findings confirm that Homo sapiens were responsible for creating this particular technology and that they had ventured as far north as the location where the artifacts were found. This establishes these early Homo sapiens as some of the earliest inhabitants of Europe. In 2018, while pursuing her PhD at the Max Planck Institute for Evolutionary Anthropology, MPI-EVA, in Leipzig, Zavala embarked on a significant project led by Jean-Jacques Hublin, the former director of the institute and a professor at the Collège de France in Paris. According to Hublin, the Ranis cave site presents compelling evidence of the initial migration of Homo sapiens across the colder regions of Europe. Surprisingly, the stone tools previously attributed to Neanderthals were actually part of the early Homo sapiens toolkit. This discovery fundamentally alters our understanding of this time period. Homo sapiens had already reached northwestern Europe well before the disappearance of Neanderthals in southwestern Europe. Zavala, in charge of the genetic analysis, examined bone fragments from both recent and older excavations at Ranis, spanning from 2016 to 2022 and the 1930s respectively. Due to the highly fragmented nature of ancient DNA, she utilized specialized techniques to isolate and sequence the mitochondrial DNA, mTNA, inherited exclusively from the mother. The identification of the skeletal remains as Homo sapiens has been verified. It is worth noting that multiple fragments exhibited identical mitochondrial DNA sequences, despite being discovered in separate excavations. This suggests a connection between the fragments, either belonging to the same individual or their maternal relatives. thereby linking the recent discoveries to those made several decades ago. Dorothea Mylopotamitakis, a former member of MPI EVA and current doctoral student at the Collège de France, utilized the field of paleoproteomics to initially determine that the bone fragments were of human origin through the analysis of bone proteins. Through the examination of Ranis mitochondrial DNA sequences in comparison to mtDNA sequences from other Paleolithic sites in Europe, Zavala successfully created a genealogical chart depicting the lineage of early Homo sapiens throughout the continent. The majority of the 13 fragments from Ranis exhibited remarkable similarity to one another and unexpectedly bore resemblance to the mtDNA of a woman's skull discovered in a cave at Zlatý in the Czech Republic, dating back 43,000 years. The only exception aligned with an individual from Italy. Zavala, an expert in studying DNA extracted from ancient bones, bone tools and sediment, expressed curiosity about the connection between the individuals in question. Were they part of the same population? What kind of relationship could they have had? She pondered. However, Zavala noted that mitochondrial DNA only provides insight into the maternal side of the story, and nuclear DNA would be necessary to delve deeper into the matter. During her examination of sediment from different layers of the Ranis excavation, Zavala discovered DNA from various mammals, but none from hominids. By combining this analysis with examinations of bone fragments using morphological, isotopic and proteomic techniques, a comprehensive understanding of the cave's environment and the diets of both humans and animals over the centuries emerges. The bones of reindeer, cave bear, woolly rhinoceros and horse found within the cave provided evidence of frigid climate patterns akin to those found in Siberia and northern Scandinavia today, characteristic of steppe tundra. These bones also suggested that the human diet primarily consisted of sizable land-dwelling creatures. Ultimately, the researchers determined that the cave served as a hibernation spot for cave bears and a denning area for hyenas, with humans only sporadically inhabiting the space. According to a publication in Nature, Ecology and Evolution, the archaeological signature found at this site, characterized by lower density, aligns with other Lincolnbian, Ranesian, Jersmanowitian sites. The most plausible explanation for this pattern is the hypothesis that small, mobile groups of pioneer H. sapiens made expedient visits of short duration. According to Sarah Pedazani, a postdoctoral fellow at the University of La Laguna in Spain who conducted the paleoclimate study, this discovery indicates that even the early Homo sapiens groups that migrated across Eurasia possessed the ability to adapt to extreme weather conditions. It was previously believed that the ability to withstand cold climates did not emerge until much later, making this finding both intriguing and unexpected. Excavations at the Ilsenholer site, also known as the Ranis site, were primarily conducted from 1932 to 1938. The leaf points discovered at this location were later determined to date back to the later stages of the Middle Paleolithic era, spanning from approximately 300,000 to 30,000 years ago. Alternatively, they may have originated from the early stages of the Upper Paleolithic period, which commenced around 50,000 years ago. Recognizing the significance of the Ranis site in unraveling the LRJ technocomplex and the shift from the Neanderthal-linked late Middle Paleolithic to the Upper Paleolithic of modern humans in Central Europe, Hublin and his research team made the decision to conduct a fresh excavation of the site, employing state-of-the-art archaeological tools. By delving deeper into the excavation site, reaching approximately 8 meters below ground level, the recent dig expanded its scope. This endeavour required the removal of a large rock which had obstructed previous excavation efforts, most likely due to its descent from the cave ceiling. Within this newly explored area, Hublin's team made significant discoveries, including fragments of flint tools and a quartzite flake that aligned with the LRJ technocomplex. Through subsequent proteomic analysis of numerous bone fragments retrieved from the site, it was confirmed that 4 of them originated from hominids. In contrast, during the excavations conducted in the 1930s, a total of 9 bone chips were unearthed and identified as belonging to hominids. Through DNA analysis, Zavala unequivocally established that the 13 bone fragments originated from Homo sapiens. To further piece together the timeline of the site, the team conducted radiocarbon dating on both human and animal bones found in various layers. Their focus was primarily on bones exhibiting signs of human modifications, as these provided a direct connection to human activity within the cave. Another first author, Helen Fewless, who is currently an EMBO postdoctoral fellow at the Francis Crick Institute in London, emphasized the exceptional correlation between the radiocarbon dates obtained from Homo sapiens bones in both excavation collections and modified animal bones from the LRJ layers of the new excavation. This strong connection solidifies the association between the human remains and LRJ. The available evidence strongly suggests that Homo sapiens intermittently inhabited the site as early as 47,500 years ago. Tim Schuler, from the Thuringian State Office for the Preservation of Historical Monuments and Archaeology in Weimar, Germany, noted that the findings from the Ilsenhöhle in Ranis have significantly altered our understanding of the chronology and settlement patterns in Europe north of the Alps. Co-leading the Ranis excavation alongside Hublin, Schuler and Weiss, Shannon McFerrin of MPI EVE and co-first author Marcel Weiss of Friedrich Alexander Universität Erlangen-Nürnberg are among the other co-authors of the Nature paper. Furthermore, Zavala, who is also a co-first author of the Nature paper, collaborated on the two papers published in Nature, Ecology and Evolution. The Max Planck Society provided financial support for the excavations and a significant portion of the subsequent analysis. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.